name is Weston Struwe. I'm a PI and a UKRI Future Leaders Fellow in the Department of Chemistry, and I'm based in the Kavli Institute. So our work focuses on sugars, complex oligosaccharides that are commonly referred to as glycans. And you can think of these as sort of very large branch structures, like a tree. But our work principally focuses on viruses and understanding how proteins on the surface of viruses are modified with these carbohydrates. And we try to understand how structure and structural dynamics regulates how these sugar structures are added. And then ultimately the interplay between dynamics with a biomolecular interaction that are largely carbohydrate dependent. And this can be with an antibody, an immune cell, a carbohydrate binding protein on immune cells, and host receptors. So the methods that we use vary, but they're largely based on mass spectrometry. So this is an eye mobility mass spectrometer, and we use this in different ways. One of the methods that we do is glycomics and glycoproteomics. So this is how we analyze the structure of carbohydrates as well as the position on a protein. This instrument's unique in the sense that it has an ion mobility cell, which separates ions based on their size, shape, and charge. And that's particularly useful for us because, as I said earlier, carbohydrates, if you think about them like branch trees, have different shapes. So we can separate these different shapes inside the instrument, and once we've separated them, we can analyze what their structures are by breaking them apart. So you can kind of think of that as a puzzle, or putting a puzzle back together from its broken pieces. So one of the samples that we've been analyzing is a biotherapeutic that's based on a linear oligosaccharide. And you can see that there are at least 3 peaks for the different isomeric structures. So it's the same structure but in different positional orientations. And then we can use the mass spectrum shown here of the 3 major peaks to figure out which one belongs to which spectrum, and then figure out what the structures are. So the Kavli is an ideal environment for many of us, particularly those of us in the chemistry department who are here. We've learned a lot in the brief period that we've been here, and it's a brilliant place for us to be. Looking forward to the collaborations, learning about new methods being developed by other groups. So I think it's very exciting to be here.